Next presentation is on microbes, a trend of flow by Dr. AMBD Alahakon, Senior Registrar in Internal Medicine, Professorial Unit, Peradeniya. Over to you, Dr. Alahakon.
Good afternoon. Thank you, sir, for your kind introduction. And thank you, CCP, for giving me this opportunity to present on this platform. When I first received the mail asking to present in your physician forum, I was thinking, what should I present? Possibly what topic would capture the attention of this audience? So I thought, why not something that has been kept like generations of physicians on their toes for centuries? So as general physicians, I think major bulk of our ward rounds is tackling infections. So we are conditioned to see microbes as our foes, as our enemies. But let me change your mind regarding that today. I will tell you that they are our friends as much as they are our enemies. So my talk today will be covering the fact what is microbiome and what is new, because this is not a novel concept. This came in 17th century. So why am I talking about it today? So I'll be covering what is new as well. And then why am I covering it in this physician forum? What is the clinical importance? Is the composition and the dynamics of microbiome can cause health and disease in patients. So we'll be addressing that as well. And then can we change our microbiome to prevent, predict, or treat diseases? That also we'll be discussing. And then we'll be discussing what is unknown and what is yet to be covered during next decade to come. Moving on, so what is microbiome? So microbiome is the collective genome of all the microorganisms, bacteria, viruses, parasites, non-pathogenic, pathogenic, both symbiotic bacteria and pathogenic bacteria living in or around all vertebrates. So when it comes to humans, it is called human microbiome. So every human acquires this microbiome before birth, after birth, and during our lifetime, during different environmental exposure, the microbiome changes. So there will be individual diversity between these microbiomes. So what is mine is not what is yours. So it's a complete different parallel civilization that we have inside me and inside you. And this microbiota diversity is not uniform throughout our organs as well. We have skin microbiota, we have gastric microbiota, GI microbiota, genitourinary microbiota, which are different from each other. So, ladies and gentlemen, I'll be not exaggerating when I say us humans are mostly microbes. We have over 100 trillions of them living in and on us. Actual fact is the microbe cells, they outnumber our cells, host cells, 10 to 1. And if you actually lose all your microbiome, you will be actually losing 5 pounds. It is that much significant. So if you can see, human is made out of visible organs, and then you have this microbiome inside and on humans, and actually you can call it a separate organ as well. And then you'll have functioning human being. So as I told you, this is not a novel concept. This concept was brought forward in early 17th century. When now people wanted to culture pathogenic bacteria, they saw that some non-pathogenic bacteria were also cultured. But culturing takes some time. It's time consuming, technically not very feasible because of these different environmental factors. So the research on human microbiome took very baby steps during the last couple of decades. But when we entered the omics era in early 19th century, and mainly in late 19th century, everything changed. So what is this omics era? Omics is basically what the suffix that we give for this gene analyzing or protein analyzing sequencing. So when you are analyzing a gene, when you know how to do PCR and get a microorganism analyzed and typed, you call it genomic analysis. And when you know about the protein, and if you can get certain mRNA pattern isolated, you call it proteomic analysis. And when you know a certain metabolite, which is secreted by one of the organisms, and when you get that analyzed, that is called proteomics. So transition from pre-omic era to omics era has changed every research in biologics and human being drastically. And human microbiome, on the other hand, has had an exponential growth in research and studies throughout this last couple of decades. So what is already known? So with this advancement of research, we know what is the composition of the microbiome. So as you know, there's something called Human Genomic Project. And along with, they have initiated, the National Health Trust has initiated Human Microbiome Project to type and analyze the human microbiome. And it ran in two phases. The first phase, they identified the microbiome using multiomics. And as you can see, there are a lot of bacteria and viruses as well, confined to different ecological niches. The gut microbiota, lung microbiota, skin microbiota. But what we need to know is that they are different from each and every individual. And the second thing that they analyzed was, it's not just the composition, because you and I might have more or less similar microbiome, but we act differently. My health is not your health. The dynamics of the host and microbiome is different. So they analyzed what affects the composition of the microbiome and what affects the dynamic of the microbiome of the host. And as I have actually summarized the findings which were carried out by this Human Microbiome Project. And as you can see, gut microbiome is the largest and the most diverse so far. So if you can see, certain host factors affect the composition and the dynamics of the microbiome. As you can see, non-specific factors like IgA and anti-metabolic proteins, mucus, they would either adversely or beneficially affect our microbiome. And there are specific factors as well. Our genome would carry certain miRNA molecules that would secrete certain proteins which might act as anti-metabolic for this particular healthy microbiome task. And then age and population would also, environment would also affect our microbiome composition. And the delivery pattern. So if I give you an example, an infant who is born vaginally would acquire the lung and skin microbiota from mother's vagina. These are healthy bacteria, mostly healthy bacteria. So they would have better immune system and they would have less asthma. The research has shown that they would have less asthma as children. Whereas the babies who are born through cesarean section, they acquire their skin microbiota mainly from mother's skin, which are basically skin microbiota. Most of them are pathobiont. Pathobiont means they are not completely healthy. If you're given a chance, they can induce a pathological process in you. There's a potentially pathologic microbiome. And the diet. And this is why we ask to breastfeed the infants. When you breastfeed the infants, the formula, the breast milk, they would have lactobacilli, mainly as the breast milk composition. And this lactobacilli would the breast milk would contain oligosaccharides, and the lactobacilli would act on oligosaccharides and it would boost the immunity by producing IgG. Whereas the formula-fed babies, the formula mainly includes enterococcus, which is not as good as lactobacilli. So as you can see, diet in infants can affect the microbiome. So I think I don't have to say diet in us, what is the impact it has on gut microbiome. It has been shown that artificial sweeteners, processed meat, they can increase the number of pathobiont and they can increase the activity of pathobiont, potential pathogenic microbiome, and reduce the activity of non-pathogenic microbiome. And antibiotics. The antibiotics that we use would potentially reduce the healthy microbiome. That being said, so I explained in my previous slides that we can change the dynamics and the composition of the microbiome. So by changing the dynamics and the composition of this seemingly non-pathogenic microbiome, it can cause disease even though they're seemingly non-pathogenic. So what are the factors that can affect this symbiosis? Usually the healthy gut microbiome would help us in maintaining our nutrition by producing vitamin B12, and they would help us to maintain the integrity of the endothelium by reducing the intestinal permeability. But when this symbiosis is broken, when it is dysregulated, there will be a dysbiosis. So what can cause this dysbiosis? As you can see, genetic background can cause a dysbiosis. That's why inflammatory bowel disease has a genetic component. And certain infections and inflammation, certain streptococcal infections can trigger certain infections, because those would act with our healthy microbiome, and there will be a dysbiosis. And our lifestyle habits, stress, smoking, certain diet, and more importantly, other environmental factors like high sugar, low fiber diet, artificial preservatives, and xenobiotics. We over-the-counter misuse all the antibiotics and probiotics and prebiotics. That is actually confusing to our human microbiome. So what we need to know is that we are confusing our human microbiome. So we are getting the symbiosis into a state that the human microbiome would go into a dysbiosis and induce diseases in us. That brings to me, what are the clinical implications of these microbiome? The research has shown that cardiovascular diseases are actually related to dysbiosis of microbiome. The vascular endothelial growth factors would increase because of the cytokines and the toxins produced by this dysregulated microbiome, and they'll be accelerated atherosclerosis. And cancer. And as we all know, this H. pylori came in 1994, when people actually thought, okay, probably cancer has something to do with microbes as well. So after that, there was exponential rise in microbial research regards to cancer. And then they have shown that microbiome would influence host cell proliferation, immune system, and metabolism, leading to cancer formation. And we have studied this to an extent. We have actually developed biomarkers to see that Fusobacterium nucleatum was increasingly isolated in patients with colorectal cancer. So they have developed new biomarkers to predict which people can have colorectal cancers as well. Intestinal diseases, I think the gastroenterologist is here and he'll elaborate more. The intestinal diseases and gut microbiome has a never-ending story. So what they say is, in IBD patients, they have isolated adherent invasive E. coli more in their microbiome compared to the health controls, and it would increase the intestinal permeability, and it would increase the endotoxin production, leading to inflammation and inflammatory bowel disease. And respiratory diseases, it has been shown that bronchial asthma patient has more Proteobacteriaceae compared to non-bronchial asthma patients. And several pulmonary fibrosis-related studies have shown that these specific staphylococcal and streptococcal organisms were cultured and isolated in these PF patients. And oral microbiota is also important because depending on the oral microbiota, due to its close proximity to the lung, you can have microaspirations and cause diseases. And then let me introduce you to this concept of gut-brain axis, gut-kidney axis, and gut-liver axis, which basically says gut is our new brain. Gut controls everything. So because gut has the major human microbiome, most diverse and most populated human microbiome. So through several neurohormonal mechanisms, it affects how we think and what we do. So this concept is known as psychobiome, which basically say whatever the toxins, chemical mediators, whatever the neural mechanisms would ultimately decide how we think. And sometimes it might go to an extent that it would trigger depression, anxiety, and other psychological disorders. And it would also affect HPA axis, hypothalamic pituitary axis directly, these endotoxins, so causing chronic stress. So gut is our new brain. And this is the gut-kidney axis. The endotoxins which are released by the dysregulated microbiome would increase periodontitis. By that, there will be increased vascular endothelium inflammation, and that is identified as an independent risk factor for CKD development. And liver disease, this has this was the talk of the town during NASH discussions in Bavino a couple of years back. Now NASH, usually non-alcoholic steatohepatitis, we can diagnose by ultrasound, we can diagnose by liver elastography, but there are new biomarkers for them. Certain dysbiotic microbiome can release certain biomarkers that are abundant in NASH patients, but not in healthy patients. Such biomarkers are hydroxyphenyl lactate and succinate, which is produced by bacteroid. And abundance of bacteroids is seen as an independent risk factor to develop liver cirrhosis. And at last but not least, diabetes. We thought microbes can only cause infections. Not really. Dysbiotic microbiome can induce diabetes in many different ways. And presence of certain microbiome in our gut would increase the likelihood of having a high insulin sensitivity, like Copococcus. So it is not only important in management of diabetes, but also as the etiology of the diabetes. So ladies and gentlemen, a couple of years back, we thought that if a patient is genetically predisposed to have a disease, and when the environment triggers something, we get the disease. But it is not the same. There are things that we can't see. So in a couple of years' time, if somebody asks what are the risk factors to develop diabetes, in addition to listing down, your traditional risk factors, if somebody says presence of X type of microbiome, presence of Y type of microbiome, I would not be surprised because we are heading that way with multi-omic research studies. So what is so important to know about how this dysbiotic microbiome affect our health? Because we can potentially prevent and treat them. We actually do this on our own these days also. We prescribe prebiotics, which are the substances that are used by the healthy microbiome specifically to grow. And we use probiotic, the actual healthy microbiome. And for this resistant Clostridium difficile infections, we use fecal microbiota transplantation. So these are things that we already use, but these are the potential treatment, engineering bacterial and phage therapy. Those are the potential treatment to increase healthy microbiome inside us and to reduce the pathogenic dysbiotic microbiome inside. So what is unknown then? The unknown is more than what is known. It is unknown how dynamic is the microbiome during common processes such as viral infections and pregnancy, whether it would change our microbiome. And it is also unknown that different individuals have more or less similar microbiome, but what changes the one from having the disease and the other one is not having the disease? Is there anything else to it? And the other thing is it presence of bacteroids in liver cirrhotic patient is a mere coincidence? Or is it a causative relationship? Or is it an cause-effect relationship? Is the patients who are cirrhotic would have bacteroid because of the cirrhosis, or because we have the bacteroids in the gut, did the patient have cirrhosis? So the cause effect is still unknown. So that is yet to be proven. And as I said, fecal microbiota transplantation, it has been practiced in several centers, but certain people would not respond to it. Certain people would get additional infections to it, and certain people would respond to it. So what are these individual mechanisms that would mediate the response of microbiome transplantation? Those are the things that need to be addressed. And hopefully with the speed that we are going with microbiome research, it will be answered in no time. So in conclusion, ladies and gentlemen, I introduced you to this amazing, diverse population. The parallel civilization which live among and in us, and those are of both non-pathogenic and pathogenic potential. Pathogenic potentials, microbiome is known as pathobionts, and they usually live in symbiosis with the host. They help host in nutrition, in maintaining immune system, and the host help them providing a place to stay and food to eat. But there are certain factors that would change the dynamics. And once the dynamics is changed, there will be a dysbiotic relationship leading to disease. So coming back to my question, are microbes friends or foes? As in every relationship, you need to give your best to have the best. So what I would say is, microbiomes are our friends and our foes. You just have to treat it right. So these are my references. I'm going to conclude my session with the saying from this Dr. Alizo Fasano, fancy name, fancy work. He's one of the physicians back in States who has a special interest in immunology and inflammatory bowel disease. He has made this statement: your gut is not Las Vegas. What happens in the gut does not stay in the gut. Thank you very much. And I would like to take this chance to thank my consultants and colleagues from Therapeutic, Professor Unit, and as well as my mentors so far, Dr. Sunitra Senanayaka, Dr. Anand Vichayanagar, Dr. Harendra Karunathilaka, for molding to be the person here I am today. And my colleagues, my friends, my family for their constant support, and as well as our patients for their selfless act carried us this far, all of us this far. Thank you very much.
Thank you, Dr. Alahakon, for that excellent presentation. I would like to open the presentation for questions. Questions from the physical audience.
Really, thank you for it as well. Normally, is always be presented with solution association.
Yes.
The problem is the causation.
Relationship. Yes.
The causation relationship to the problem. That's what it is.
Yes, sir.
So that will be very difficult to do.
Yes, sir.
But I agree with you that, for example, we have a lot of patients with the young men who hardly see a benefit in front policies. Now it's so common. You see, and the front policies. Then we know that convenience. Probably, but it is true. So you advocate that they are using a lot of antibiotics and advertising on the television, they clean the floors, protect the bacteria and clean yourself constantly with various lotions to clean the bacteria. So do you think that we should advocate to remove this type of behavior from advertisement from a media?
Yes, thank you, Sir, for the question. The first part of the question, sir, yes, the causal relationship is the problem. So the human microbiome project, so the phase 2 was carried out to see whether there's a causal relationship. Actually, they have concluded 3 things. First thing is IBD, diabetes, and the third thing is preterm delivery and microbiome. So in that study, they have shown that diabetes and IBD might have causal relationship because they have done a longitudinal study, but still not concluded. So they are hopefully planning for a 3rd stage to see the causal relationship. And because in the research article also, the last thing in conclusion that they are actually, they have detected the deficit and they wanted to correct it. And the second thing is, I think it's more or less a diplomatic question whether to do that. But what I would say is some things are better left untouched. So I think human microbiome is one of them because we don't know what it is actually. So it better be left untouched. So that's what I had to say. Thank you, sir.
Yeah, there's a question.
Thank you very much, Madam Chair. I think we have topic now. Now, when you talk about all the things that can cause harm,
Yes.
are there beneficial effects?
Yes, sir.
Change in the market.
Yes.
One question was why the difference in individual? Why we have different and if that's so that the disease patterns have changed individually.
Yes. Yes, sir. Thank you for that question. The first part is anyways, symbiosis, GI microbiota, every lung microbiota, everything has a beneficial effect. Like it would boost our immunity, it would help in nutrition as in vitamin production. And as you say, it's in this human microbiome project as well, they have recognized certain microbiome which is present abundantly in certain individuals, and they are more protected than the others to in the cases of getting IBD or liver cirrhosis. Like this Propionibacteria was it was thought to be beneficial in cirrhosis, something like that. So there are certain diverse group of microbiota which are thought to be beneficial, and there are certain groups that are thought to be adversely affecting the outcome. And the second question, sir, with how, what affects the diversity of the microbiome in different individual? As I said, it depends from your birth onwards. And until recently, it was thought uterus was sterile. But now we know before birth also people, like the infants get their skin microbiome and the lung MGI microbiome. So depending on their uterine environment, the maternal microbiota becomes their microbiota. That's the first thing. And then maternal vaginal microbiota becomes their skin microbiota. So from the birth onwards, depending on the place we stay, the things we do, microbiota changes. But studies have not yet proven, like because this HMP study, they only include 600 odd individuals. So we have to do large scale studies to see whether, staying in this environment, coming from the same, Colombo household, does it affect our microbiome? Does it mean that Colombo has this microbiome? So we need larger scale studies to see whether how the environment affects. So what we know for now is to affect the microbiome, it depends on your delivery, your food, your chronic stressors, and use of xenobiotics as well. Thank you, sir.
Thank you very much, Dr. Lahakon, for the presentation. In absence of any other questions, I would like to hand over the certificate of appreciation on behalf of the Silot College of Physicians for your contribution today.